All right, our next speaker in the session is Magdalena Russell from the Fred Hutch.
All right. Hi, everyone. Thank you, first of all, to the organizers for having me today. I'm excited to be here and talk to you about a project that I've been working on with my advisors, Phil Bradley and Eric Mattson, both at Fred Hutch. And we've been combining genotypes and T-cell receptor distributions to infer the genetic determinants of VDJ recombination probabilities. And just to give a breakdown of what I'm going to be talking about today, I'm going to start with an overview of T-cell receptor generation, including a brief review of VDJ recombination and the actual enzymes involved in the process. And then I'll dive into the main question that we're interested in, which is how does transgenetic variation impact VDJ recombination, and what does that tell us about the underlying process? And specifically, I'll focus on two stories that we've been working on. The first is where I'll spend most of our time, which is our work to identify genetic variants using genome-wide association inference. And then I'll kind of hint at where we're going next, which is to try to place our results into a mechanistic context using model-based statistical inference. So for the sake of my talk today, I want us to use a water pipe as an analogy for TCR repertoire formation. And we can think of the contents of this pipe as being influenced by generation, selection, and exposure history. As each TCR is generated, it is spit out into the water pipe. And as I'm sure you all are very familiar, most TCRs in humans are composed of two protein chains, an alpha chain and a beta chain. And in the beta chain specifically, a single V gene, D gene, and J gene will be randomly chosen to be recombined together from the pool of germline genes within the beta locus. And as those genes are joined together, they're joined in this intentionally random way, intentionally messy way that ultimately promotes diversity of the recombined locus. But I want to dive into how that joining process actually occurs, because it'll become really important as I share my results later on. So in this example, I'm showing a V gene being joined with a D gene. And the first step in this process is that the intervening chromosomal DNA between each gene is removed to form hairpin loops at the ends of each gene, as you can see there. And this is done by the RAG complex. Next, these hairpin loops are nicked open, often asymmetrically, by the Artemis DNA PKCS protein complex to create those single-stranded overhangs at the ends of each gene. And those overhangs are typically 4 nucleotides in length. 2 of those nucleotides will be palindromic nucleotides, or P nucleotides. From here, nucleotides may be deleted from the gene ends through an incompletely understood mechanism that's suggested to involve the Artemis protein from earlier. And this deletion can remove traces of those P nucleotides from before or actual regions of the gene sequence. Next, non-template encoded nucleotides known as N insertions can be added to the gene ends by the enzyme terminal deoxynucleotidyl transferase, or TDT, as I'll be calling it for short. And from here, once the nucleotide deletion and addition steps are completed, the strands can be paired, if possible, and ligated together to complete that joining process. This same mechanism is repeated for all of the junctions that need to be joined to eventually complete the receptor that will go on to become a TCR. So back to our water pipe analogy, we can think of this TCR generation process as the water source that's continuously spitting out these random TCRs into the pipe. And then following generation, positive and negative selection can act as a sort of filter within the water pipe, shown in blue. And exposures can act as sort of water pipe expanders by increasing the frequency of various TCRs in the repertoire, shown in red. And so over time, the repertoire may reflect biases of each of these 3 processes: exposure history, selection, or generation. And now all 3 of these processes are extremely probabilistic. But traditional molecular biology experiments have really taught us a ton about them. But those experiments come with a few limitations. The first of which is that we can't do knockout experiments in humans, and so that makes it challenging to learn about these mechanisms from an in vivo perspective. And second, it's difficult to get actual parameter estimates that describe the outcomes of these probabilistic processes. So we can't think quantitatively when we learn about how these mechanisms are working. But that's where repertoire data comes in. We can sample an individual's repertoire at that stage in the water pipe shown in green, and combine that data with statistical models to try to understand or gain a basic understanding of these processes. And as I'm sure you're very familiar, this process has been very successful previously, and the AIRRC community has done a lot of work in this area. But what we're interested in is how we can combine repertoire data with statistical models to inform a mechanistic understanding of the TCR generation process. So we can start thinking about questions like, how does the mechanism vary across individuals or in the context of genetic biases? And so that brings me to this main question that we've been interested in, which is, how does transgenetic variation impact VDJ recombination? And what does that tell us about the underlying process? Now, there has been a lot of work in this area to try to explore this relationship between genetic background and how it can influence TCR generation. Studies in monozygotic twins have reported similarities in VDJ gene usage and in insertion lengths, but also repertoire similarity in response to acute infection. Other groups have shown that variation in the MHC locus can bias TCRB gene usage within the post-selection repertoire. And so while this work clearly illustrates that genetic similarity implies repertoire similarity, the extent to which specific variations are associated with biasing VDJ gene usage or VDJ recombination probabilities has not been fully explored. And so given this knowledge, that's what we set out to do, identify these variants using genome-wide association inference. But before I discuss the results from that work, I want to discuss this method of genome-wide association inference in general. GWAS, as you've probably heard it called many times, aims to identify associations between a genetic variant and some observable trait. In this example, I'm using height, by testing for differences in allele frequencies between individuals who are ancestrally similar, but who differ phenotypically in the context of the trait. And we often visualize these results in a Manhattan plot, as I've sketched below, where we plot the chromosome position on the x-axis with the negative log 10 p-value of the association on the y-axis. So in this example, each green dot represents an association between a single variant and the trait, which is height here. And then we define a significance threshold, shown by the gray line, where all associations above that line are considered significantly associated, but all associations below the line are not. And this significance threshold allows us to control our false positive rate. So the position of that line will increase as we increase the number of variants that we're testing. So for our work, we want to infer associations between genetic variants and inferred TCR repertoire features, such as the extent of deletion, the number of N insertions, or the V gene, D gene, and J gene usage frequency. And to do this, we use data from around 400 individuals from those two papers highlighted below that pairs TCR beta repertoire sequencing and a large SNP array data set. And additionally, we can measure TCR repertoire features separately between both productive and non-productive sequences. And if we think back to our water pipe analogy, the TCR generation process is constantly spitting out both non-productive and productive sequences. But because non-productive sequences do not generate proteins that participate in the selection process, we can think of them as not passing the selection filter. And so later on in the pipe, towards the pink spot, we will only be observing productive sequences in addition to non-productive sequences, which are hitchhiking within a T cell that has a productive rearrangement. So as we measure these associations between genetic variations and TCR repertoire features, we want to do this separately for productive and non-productive sequences, because non-productive sequences should reflect the statistics of the generation process, whereas productive sequences should reflect the statistics of both generation and selection. But as we started this analysis, we realized that population structure variation within our sample cohort could be confounding our associations. As I alluded to earlier, GWAS relies on the population being ancestrally similar to avoid confounding this relationship between genotype and phenotype. And so here we've taken all of the genotypes from the data set that we have and projected them into PCA space, and then clustered individuals based on their PCs into what we call ancestry informative PCA clusters, which are reflected in the colors shown in the bottom plot. And so you can see a lot of that cluster represented in the various colors, where in PC1, on the x-axis, you can see that the Asian-associated group is clustering out towards the bottom in orange, whereas in PC2, you can see the African-associated PCA cluster clustering out towards the top, shown in kind of that turquoise color. And you can see the proportion of variance explained by these PCs on the top plot. And so with these clusters, we found that various genotypes and phenotypes actually varied significantly across them. In the context of N insertions, for example, we found that the extent of N insertion actually varies by ancestry informative PCA cluster. Where on the x-axis, we're plotting PCA cluster, and on the y-axis, we're plotting mean total N insertions within each individual's repertoire. And we found that in productive sequences, the Asian-associated group had significantly fewer mean total N insertions than the population mean. And additionally, we also found that SNP minor allele frequencies varied by ancestry informative PCA cluster, where again, we're plotting the PCA cluster on the x-axis and the allele frequency of a subset of SNPs, which we later went on to find to be associated with lower levels of N insertion. And we found that the Hispanic-associated PCA cluster and the Asian-associated PCA cluster had significantly higher mean allele frequencies compared to the population allele frequency. And so because of this potentially confounding relationship between genotypes, repertoire features, and population structure, we realized that we needed to correct for population stratification in our model design. To do this, we can include a subset of those PCA scores that we showed, or that I showed earlier, for each individual as additional covariates within our model. And we're doing this sort of correction for all of the GWAS results that I'll be showing today. So I want us to start by looking at the results, looking for associations between genetic variants and the gene usage frequency of various VD and J genes in the TCR beta locus. And I'm going to show a Manhattan plot where the gene usage frequency associations are all kind of piled onto one plot, but they're really representing the association with individual V genes despite being colored all together in the same color. So with this approach, and using a conservative significance threshold, we identified around 9,000 significant associations genome-wide for non-productive and productive sequences. And of these associations, around 7,000 were located within the TCR beta locus for the gene usage frequency of various VD and J genes. But we also identified around 1,300 significant associations between V gene usage and genetic variants located within the MHC locus, within productive sequences only. And now that observation, as I talked about earlier, has been noted previously, but here we noted that we did not observe those associations within non-productive TCRs. However, that's not overly surprising given the known relationship between V genes and MHC during the selection process. So we also use these methods to identify associations between genetic variants and the extent of N insertions. But before looking at these associations on the genome-wide scale, we first wanted to look at the relationship between the extent of N insertions and genetic variants located within the gene encoding the TDT protein, due to its known direct involvement in actually adding N insertions during VDJ recombination. And this here is one of the first papers that actually made that mechanistic link. And so when we restrict our analysis to the DNTT locus, which is the gene that encodes the TDT protein, we identified around 230 significant associations between the extent of VD and DJ N insertion and genetic variants within these loci for both non-productive and productive TCRs. But we found that the magnitudes of these effects were actually larger for non-productive TCRs as opposed to productive TCRs. And when we expanded our analysis genome-wide, only 3 of these associations remained significant. You can see those on the top plot within the DNTT locus region. And interestingly, we didn't really notice any other associations at other regions in the genome. All right, so lastly, and I think most excitingly, we also use these methods to identify associations between genetic variants and the extent of nucleotide deletion. But it had been noticed previously that the extent of deletion actually varies by gene usage. And our earlier results identified many genetic variants which were associated with biasing gene choice. And so it became really clear that in order to directly measure the association between genetic variants and the extent of deletion, we needed to correct for gene usage in our model design, in addition to existing correction for population stratification. And so when we did this, we actually identified a lot of not interesting associations between 5' gene trimming and genetic variants within the TCR beta locus. We believe these are due to a correlation between SNP genotype and TRBD2 allele genotype, as opposed to some relationship that's actually related to trimming. But more excitingly, we did note around 70 highly significant associations within the DCLRE1C gene locus, which is the gene that encodes the Artemis protein that we discussed earlier. We noted these associations for both V gene and J gene trimming of productive and non-productive sequences. But again, the magnitudes of these effects were larger for non-productive sequences as opposed to productive sequences, which might suggest some sort of TCR generation related bias as opposed to one that's isolated just to the selection process. And if we zoom in on all of the significant Artemis associations from that previous plot and compare their Artemis gene locus position with the association type on the y-axis, and now color each association by the strength of the p-value for the association, we can see that many of these associations actually overlapped between trimming and productivity types, and were located within non-coding regions of the gene, where you can see the exons and promoter locations noted on the bottom in green and red. And that remained true for even the most significantly associated variants that you can see in darker blue towards the center of the plot. But we did notice one non-synonymous association that was significantly associated with biasing the extent of V and J gene trimming for both productive and non-productive sequences. And you can see that with the arrow pointed also towards the middle. And we noticed this association both within this primary cohort and within a second independent validation cohort as well. But I think zooming out when looking at these results, I think it's important to note that the actual involvement of Artemis in the trimming mechanism has not been confirmed. And so despite not necessarily identifying a causal variant, our results do suggest that Artemis could be the nuclease actually responsible for trimming during VDJ recombination, which I think is exciting. All right, so that brings me to the conclusions for this portion of my talk. I hope I've illustrated that we've precisely mapped transvariation, which leads to different VDJ recombination probabilities. We found some interesting population structure variation. We found an independent line of evidence to support that Artemis may be the nuclease actually responsible for trimming during VDJ recombination. And otherwise, we sort of found signals where you would expect them, and we didn't really find any signal outside of where you would expect. And if you're interested in more details, I'd encourage you to check out our recent paper in eLife shown there. All right, so with my last few minutes, I want to sort of hint at where we're going next, which is to try to place those results from the associations that we identified within the Artemis region that were associated with changing the extent of trimming, trying to place those into a mechanistic context using model-based statistical inference. And to do this, I sort of alluded to earlier that traditional molecular biology experiments have provided a lot of really powerful insights into both how the trimming mechanism works and how Artemis functions as a nuclease outside of VDJ recombination. For example, Artemis has been found to recognize DNA structural features such as single-stranded to double-stranded DNA transitions, but not necessarily recognize specific DNA bases. And so we can take that mechanistic understanding and combine it with big-ish data sets of repertoire data and statistical inference to fit probabilistic models that can predict the probability of trimming at a certain site given a gene sequence and certain mechanistically inspired parameters. We can then use various model evaluation metrics to sort of revise our mechanistic understanding and maybe come up with different mechanistically inspired parameters and fit new probabilistic models and continue on in this cycle as we continue revising our mechanistic understanding. But I think maybe even more powerfully, we can then get out parameter estimates for our mechanistic models. So we can start to think about mechanistic understanding in a quantitative way. So maybe certain parameters could be more important or have more relative importance than other parameters, which could help us continue learning about how the mechanism is working. And then we can look towards comparing those estimates in the context of some of those genetic variants, which we identified, especially within the Artemis locus, to be associated with changing the extent of trimming. So unfortunately, I don't have time to discuss the results from this work today, but if you're interested, I'd love to chat more over coffee or whatever. All right. Thank you so much. Yeah, first of all, thank you to Eric Mattson and Phil Bradley at Fred Hutch. They've been wonderful mentors in this work, as well as some great statistics collaborators, Noah Simon and Dave Levine at the University of Washington, a really generous VDJ recombination expert at Yale, David Schatz, and then Paul Thomas and his group at St. Jude's, the Nicaragua Ministry of Health, and University of Michigan for providing some really useful validation data sets for our GWAS work. So thank you, everyone, and I can now take whatever questions you have.
I'm wondering if you've looked at the base composition of and nucleotides addition and seeing like there's any genetic association with what bases get added.
Yeah, that's an interesting question, and we actually did not look at that. I guess we spent most of our time focusing on the trimming results because, yeah, TDT was very clearly the only association that we found for the number of nucleotides, but that would be, yeah, definitely an interesting thing to look at.
That very nice introduction to VDJ recombination, which is going to follow in the next talk. But I was wondering, do you have any models or thoughts about why the association with TDT or with Artemis is more on the non-productive alleles, even though, you know, afterwards it shrinks a little bit? Yeah, I
guess our thought on that is that, you know, it's maybe related to some sort of mechanistic change in the actual generation mechanism, and that perhaps, you know, so yeah, so I guess in the context of the Artemis variants, those variants are leading to greater amounts of trimming, and so maybe those sequences that have really large amounts of trimming are then getting selected out and not appearing in the productive repertoire. So maybe the associations are less strong in those.
Right. So I think you're saying exactly what I was considering when you were talking, that perhaps the extent of, you know, if you get too many N nucleotides added or too much DNA sequence removed, then that has a greater tendency to be non-productive.
Right. Yeah.
Is that, yeah?
That's how I've been thinking about it. Yeah. Yeah.
Good. Thank you.
I guess following that up, can you say anything about the magnitude of the effect size, like between these different SNPs? How many nucleotides are we talking about in terms of trimming and deletion? And
not very many, like 0.2 for trimming is the difference. But yeah, I mean, enough to be significant in a GWAS and perhaps create a functional change.
Within VJ rearrangements, the ones that are physically closer on the locus tend to rearrange more. Do you see any differences within the trimming depending on how far away the V and J are?
Oh, interesting. So Yeah, so I guess we computed all associations like, just within V, D, or J, but not like considering the associated J with the V trimming. But yeah, that would be really interesting.